Have you ever really stopped to think about it? How a single tiny cell, just one microscopic blob, transforms into, well, us, into a whole complex organism.
It is pretty mind-bending, isn't it?
Yeah, like a symphony of different parts, tissues, organs, all working together. How does that first blueprint, that single cell, get read and built upon piece by intricate piece? Seems almost impossibly precise.
It really does. And that fundamental question is what developmental biology is all about. It's the study of those incredible dynamic processes, how cells, tissues, whole organisms grow and change over time.
Yeah, okay.
And what's really exciting, and why we're talking about it now, is that the field's kind of entered a new golden age. That's what people are calling it.
The golden age, why now?
It's fueled by this amazing convergence of, well, really powerful new technologies all coming together at once.
Okay, so that's our mission for this deep dive then. Let's unpack that. Let's explore how developmental biology has evolved, the big ideas and the tech revolutions that have totally changed how we understand life's basic assembly instructions.
Sounds good.
We're digging into a really fascinating review article by Prisca Liberali and Alexander Shear, and we want to pull out the key stuff, maybe connect some dots you hadn't thought about before.
Yeah, there's some really surprising connections in there.
All right, let's kick things off.
Yeah.
To really get where developmental biology is today, I think we need a quick look back, right?
Yeah.
It hasn't always been about fancy tech.
Absolutely not. You have to appreciate the journey. It's been this incredible acceleration of discovery.
So where did it start, like before the molecular stuff?
Well, if you go back to, say, the 1970s, the field was very descriptive. Scientists were meticulously observing and describing developmental phenomena, like natural historians almost.
And watching things happen.
Watching very closely, yes. And identifying developmental mutant organisms where something went wrong, which gave clues about the normal process. A huge step then was defining C. elegans, that tiny little roundworm, as a key model system.
Ah, the worm. Why was that so important?
Because it's transparent. And crucially, it has a completely fixed cell lineage. Every single worm develops in almost exactly the same way, cell division by cell division.
Wow.
Yeah. So you could literally trace the fate of every single cell from the fertilized egg to the adult worm. It was like having a perfect, transparent map of development unfolding. A total game changer for understanding basic principles.
Okay, so from observation and mapping, what came next? You mentioned a revolution.
Right, the 80s and 90s, that was the molecular genetics revolution, a massive turning point. Researchers went from just describing to figuring out the actual mechanisms. They started isolating specific genes, figuring out the molecular pathways controlling development.
Like finding the instruction manual.
Exactly. And one of the landmark discoveries was the HOX complexes and the homeobox. This was huge.
HOX genes, I've heard of those. They're involved in body plan, right?
Precisely. Imagine finding this set of genes, this toolkit, that lays out the basic body axis, head here, tail there, or segments in between. And the truly stunning part, this toolkit is incredibly ancient and conserved across vastly different animals. Flies, mice, humans. We all use basically the same HOX genes to pattern our bodies.
That's wild. So the same fundamental instructions.
Fundamentally, yes. It showed this deep evolutionary connection, a shared language for building bodies. And importantly, it wasn't just academic. Researchers quickly saw that mutations in these key developmental genes were often linked to human genetic diseases.
Ah, the health connection.
Right. Suddenly, understanding fruit fly development had direct relevance to human health. That era really laid the groundwork for understanding gene regulation, pattern formation, organogenesis, all the core processes.
Okay, so observation, then finding the genes. What happened in the last, say, 25 years? Did things slow down?
Oh, definitely not. If anything, things diversified and accelerated again. The field expanded way beyond just embryos. People started digging into morphogenesis, how tissues physically fold and shape themselves.
Like origami with cells.
Sort of. And stem cell biology, how tissues maintain themselves in adults, evolution, regeneration, even trying to engineer organs in the lab.
Organ engineering. Wow.
Yeah. And this brings us to now this current golden age, which is really driven by a massive boost from technology.
Okay, the tech. What were the key pieces that, as you said, blew the doors open?
Several things came together. First, genomics. Yeah. Just the sheer ability to sequence DNA rapidly and cheaply. Think about it. The first multicellular animal genome, C. elegans again, was sequenced in '98.
Right, having the whole parts list.
Exactly. Suddenly you could see all the potential genes involved.
Yeah.
Then imaging. Things like confocal microscopy gave us much clearer 3D views inside cells and tissues.
Better pictures.
Much better. But then came things like light sheet microscopy. This is incredible. It lets you image a whole living embryo in 3D over time without damaging it too much.
So you can actually watch development happen live.
Yes. In developmental live imaging, you can literally track individual cells moving, dividing, changing shape in real time within the whole organism. It's like going from a blurry photo to a high-definition movie.
That's amazing. What else?
Genome editing, specifically CRISPR. This gave scientists the power to make very precise changes to DNA, to test what specific genes do during development with unprecedented ease and accuracy.
Turning genes on or off to see what
For
happens.
tweaking them, yeah. Then add computational biology and AI. We're generating unbelievable amounts of data from sequencing and imaging. You need powerful computers and algorithms to make sense of it all.
Right, handling the data flood.
And finally, new model systems are emerging, like studying tardigrades for their incredible resilience. But maybe the most revolutionary are organoids.
Organoids, mini organs in a dish, right?
Exactly. These are tiny 3D structures grown from stem cells, embryonic or adult, that self-organize and mimic the structure and function of real organs. Mini brains, mini guts, mini livers. It allows us to study human development and disease in a dish,
Right.
in ways that were just impossible before. It's a huge leap.
Okay, that's quite a trajectory from just watching to finding genes to now building mini organs and watching everything live. If you had to recap this transformation, what really stands out?
I think two main things. First, the shift from descriptive studies to uncovering the actual precise molecular and genetic mechanisms. We're not just asking what happens, but how exactly does it happen?
Okay, the mechanism focus.
Second, the incredible expansion of scope.
Yeah.
It's not just about embryos anymore. It's stem cells, regeneration, aging, evolution, tissue engineering. It touches so many areas of biology.
Got it.
And underpinning all of this, the technology explosion. Genomics, imaging, editing, computation, they're the engines driving this golden age.
So this really brings us to the core of it all, doesn't it? The blueprint itself. How are those genetic instructions actually read? How does one cell type become hundreds of different cell types all in the right place? That precision seems key.
Yeah, this is where it gets really fascinating for me. If development is like, I don't know, a huge construction project, then gene regulation must be the super detailed architectural plan, right?
Yeah.
Every cell, every worker needs to know exactly what its job is, where it needs to be, and crucially, when it needs to do its thing.
That's a fantastic analogy. And it is meticulously choreographed. At the heart of it, you've got a few key players working together in these intricate networks.
Who are the main players?
First, you have transcription factors, or TFs. Think of them as molecular switches or maybe conductors. They bind to specific parts of the DNA and tell genes whether to turn on or off or how strongly to turn on.
Okay, the switches. Then you have the DNA sequences they bind to, particularly things called enhancers. These are like docking sites or control panels for the transcription factors. They determine where and when a gene gets turned on. An enhancer might only be active in, say, future brain cells at a specific time.
So location, location, location for the switches.
Exactly. And the third big player is chromatin. That's how the DNA itself is packaged up with proteins in the nucleus. It's not just packaging. The way it's folded or chemically tagged can make genes accessible or inaccessible.
So the packaging itself controls things.
It plays a huge role, yes.
Yeah.
It adds another layer of control. And all these elements, TFs, enhancers, chromatin, work together in complex gene regulatory networks, or GRNs. These networks are the real program for development.
That sounds incredibly complex. How did scientists first start figuring this out?
Some classic experiments were really foundational, like the discovery of TF cascades. A famous example is in the fruit fly embryo Drosophila. There's a transcription factor called becoid.
Right, you mentioned that earlier.
Yes. It's not a signal between cells, but within them. It forms a gradient high concentration at one end of the embryo, low at the other. And different concentrations of becoid turn on different sets of genes.
It's like a dimmer switch based on concentration.
Exactly. It tells cells their position along the head-to-tail axis. It was a beautiful early example of how a simple gradient of one molecule could specify different cell fates and pattern the embryo. It became a classic paradigm.
Okay, so gradients can set things up. What about making specific types of cells?
That led to the idea of master regulators. These are powerful transcription factors that can sometimes almost single-handedly push a cell towards a specific identity. The classic example is MyoD.
What does MyoD do?
Amazingly, if you force a connective tissue cell like a fibroblast to express MyoD, it can convert that cell into a muscle cell.
Just like that.
Wow. It shows the power of these master switches. We also figured out more about enhancers, using reporter genes, basically, hooking up an enhancer to a gene that makes a visible color. This let researchers see exactly where and when specific enhancers were active, confirming they act as these key regulatory switches controlled by combinations of TFs.
So you could visually track the control panels working.
Precisely. And then came the realization about chromatin as memory. Discovering chemical modifications to DNA and its packaging proteins, things like methylation and acetylation, led to the idea that the chromatin state records a cell's history.
Cellular memory. How does that work?
Well, decisions made early in development can leave lasting marks on the chromatin, making certain genes permanently accessible or inaccessible in that cell lineage. It helps maintain cell identity over time. A liver cell stays a liver cell, partly because its chromatin remembers it's a liver cell.
That's a huge conceptual leap from just code to dynamic memory. How are the newer technologies refining this picture?
Oh, they're adding incredible layers of detail and complexity. For example, at the really fine-grained level, using advanced imaging like single-molecule RNA ISH.
Single molecule. Seeing individual RNA molecules.
Yes, it revealed that genes don't just turn on smoothly. They often fire in bursts, short, intense periods of activity, transcriptional bursting.
Like flicking a switch on and off rapidly?
Kind of. And the regulation is subtle. Those TF binding sites on enhancers, often they have quite low affinity, meaning the TF doesn't stick very strongly or easily.
Wouldn't that make it unreliable?
You'd think so, but it seems to be a feature, not a bug. It means that gene activation often requires multiple TFs binding cooperatively or very specific TF concentrations. It makes the system highly sensitive and specific, preventing accidental gene activation. It ensures precision.
Ah, like needing multiple keys to turn it once.
Good analogy. And then, zooming out, the genome-wide technologies have been transformative. Single-cell RNA sequencing, single-cell ATAC-seq, which maps accessible chromatin.
Looking at individual cells' activity.
Yes, across thousands or millions of cells at once. This lets us build incredibly detailed atlases of development maps, showing all the different cell types, their gene expression profiles, and how they change over time and space. We can trace developmental trajectories.
Like Google Maps for cell development.
Exactly. And even without manipulating cells, by combining these multi-omics data with computational predictions of where TFs bind, we can start to infer the likely gene regulatory networks controlling these processes.
So you can predict the wiring diagram.
Yeah, we're getting there. A fascinating example came from studying the thymus, the organ where T cells mature. Single-cell analysis showed how certain thymus cells actually borrow TFs, usually used for other cell types, to basically show developing T cells what self looks like, helping eliminate potentially autoimmune T cells.
Wow, using the enemy's playbook, sort of?
In a way. And AI and deep learning are pushing this even further. People are training neural networks on genomic data to predict where TS will bind, which enhancers will be active, even reconstructing GRNs. We're getting close to being able to define and even design regulatory elements computationally.
Design genetic switches in a computer.
It's heading that way.
Yeah.
For instance, designing enhancers that are only active in specific cell types. And we're also learning about the whole genome structure in 3D. It's not just a linear string.
How it's folded up matters.
Immensely. Techniques like chromosome conformation capture show the genome is organized into distinct neighborhoods called TADs, topologically associating domains. Interactions usually happen within a TAD. And amazingly, in the Hox complex, specific DNA loops that form and break have been shown to act like a developmental timer, controlling the sequential activation of Hox genes. The physical folding itself is part of the clock.
Mind-blowing. The precision is just staggering. From bursts to 3D folding timers, what's the big takeaway here for you?
It's the multi-layered nature of the control, from the fine biophysical details of TF binding and bursting to the global organization of the genome in 3D, all working together to ensure robustness and precision. And the fact that we can now see and start to predict this complexity is just incredible. The potential for understanding basic biology and for medicine is enormous.
Which brings us perfectly to the sickle cell anemia therapy. That's a really powerful example of this, isn't it?
It's a landmark case. Absolutely. It shows the direct line from fundamental research on gene regulation to a life-changing therapy.
Walk us through how that worked.
Okay, so researchers studying how hemoglobin genes are regulated specifically, how we switch from fetal hemoglobin before birth to adult hemoglobin after discovered a key repressor protein called BCL11A.
A repressor, so it turns something off.
Exactly. In adults, BCL11A turns off the fetal hemoglobin gene. The problem in sickle cell is the adult hemoglobin is faulty. But fetal hemoglobin works fine. It doesn't sickle.
So if you could turn fetal hemoglobin back on,
Precisely. The idea was, what if we could reduce BCL11A levels, specifically in blood stem cells? That should allow fetal hemoglobin production to restart.
and how did they do that? That's where CRISPR came in.
Yes, they didn't target the BCL11A gene itself, but a crucial enhancer that controls BCL11A expression only in red blood cell precursors. Using CRISPR, they made a tiny, precise mutation in this enhancer in the patient's own blood stem cells.
Just tweaking the switch, not the whole gene.
Exactly. This lowered BCL11A levels just enough, fetal hemoglobin turned back on, and it dramatically reduced sickle cell symptoms in clinical trials. It became the first approved CRISPR-based therapy for a genetic disease.
Wow. A direct result of understanding those enhancers and repressors.
Absolutely. A beautiful illustration of the medical power locked within developmental gene regulation.
Okay, so we've got the blueprint being read with incredible precision through gene regulation. But how do all these cells, following their individual instructions, coordinate to build complex shapes and patterns? How do they form an arm or a gut tube? That seems like a whole other level of organization.
It is. And that brings us to pattern formation and morphogenesis.
Right, it's like imagine building a city. You can give every worker perfect instructions, but how do they coordinate to build the roads, the districts, the skyscrapers in the right places relative to each other? It feels like self-organization on a grand scale.
That's a really good way to think about it.
Yeah.
Pattern formation is essentially cells acquiring different identities based on their position, arranging themselves into functional tissues and organs. And often, this involves signaling between cells.
Cells talking to each other.
Yes. A key concept here is the morphogen. These are signaling molecules that spread out, often forming a concentration gradient. Cells sense the local concentration of the morphogen, and that tells them where they are and what identity to adopt.
Like the bicoid gradient, but this time the signal travels between cells.
Exactly. It provides positional information. Think of it like a chemical GPS signal, telling cells their coordinates within the developing tissue.
How do these signals spread? Just diffusion?
Diffusion is common, yes. But we're finding other cool mechanisms, too. Sometimes signals are passed along via long, thin cellular projections like little fingers reaching out called filopodia or cytonemes.
Wow. Direct delivery service.
Sort of. Or signals can be transmitted through relay mechanisms where a cell receives a signal, gets activated, and then produces the same signal, passing it on to its neighbor, like a chain reaction.
Okay, more complex than just diffusion. And can this create complex patterns, like stripes or spots?
Definitely. Simple gradients are one thing, but you can get much more intricate patterns. For example, combining local self-activation of a signal with long-range inhibition, where the signal turns itself on nearby but inhibits itself further away, can generate Turing patterns.
Turing patterns, like on animal coats, zebras, leopards?
Precisely those kinds of patterns. It's a mathematical concept that plays out biologically. Or you can have feedback loops that create oscillations in time, like the segmentation clock.
A clock, what does that do?
In developing vertebrates, there's this oscillating network of gene expression that sweeps across the tissue like a wave, rhythmically laying down the precursor blocks for vertebrae and ribs. Tick-tock, a new segment forms.
Like a biological clock carving out the spine.
Yeah.
Incredible. Are these classic ideas holding up as we get better tools to test them?
That's a great question. The new tools are definitely allowing us to test assumptions much more rigorously. Take optogenetics, for instance.
Using light to control proteins.
Yeah, you can engineer cells so that you can turn a signaling pathway on or off, or move a protein around just by shining light on it. This lets researchers test, for example, if a smooth spatial gradient is truly required for patterning, or if maybe just short-range interactions or even the dynamics of the signal are enough. You can perturb the system with incredible precision.
So really poking and prodding the system live.
Exactly. Now, moving from the pattern of cell identities to the actual physical shape. That's morphogenesis, how tissues bend, fold, stretch, and move.
Yeah, the actual construction process.
It's a dynamic interplay between what individual cells do and how the whole tissue behaves mechanically. A really big idea here was differential adhesion.
Cells sticking together differently.
Yes. The idea, proposed decades ago, is that cells with different adhesion properties will sort themselves out, like oil and water separating. If cells prefer to stick to similar cells, they'll tend to clump together, forming boundaries. The discovery of cadherin's key cell adhesion molecules really validated this.
So stickiness helps define tissues.
A lot. And now we know it's not just about which adhesion molecules are present, but also about the physical forces involved, tension at the cell surface, the cell's internal scaffolding, the actin-myosin network.
The cell's muscles.
Exactly. Then there's cell migration. Cells have to move, sometimes individually, sometimes in large groups.
How do they know where to go?
They follow guidance cues, chemical gradients, chemotaxis, gradients of stickiness on surfaces, haptotaxis, physical cues like stiffness or channels, mechanotaxis, durotaxis, even electrical fields can guide them.
Lots of different road signs.
Definitely. A classic example of individual migration is primordial germ cells, the precursors of sperm and eggs navigating through the embryo, following a chemokine signal to reach the developing gonads.
Like finding their way home.
Yeah. And for collective migration, neural crest cells are a fantastic model. These cells migrate in streams away from the developing spinal cord to form nerves, pigment cells, parts of the face.
They travel together.
Often, yes. And they coordinate their movement using things like planar cell polarity, PCP signaling. This pathway helps align the polarity of cells within the plane of a tissue so they can all move in a coherent direction. This drives large-scale movements like convergence and extension, where a tissue narrows and lengthens, crucial for shaping the body axis.
So they coordinate their internal compasses. What about the stuff between the cells, the matrix?
The extracellular matrix,
Yeah.
ECM. For a long time, people thought of it as just inert scaffolding, like biological concrete.
But it's more than that.
Much more. We now know the ECM is incredibly dynamic and instructive. Its composition changes constantly during development. It provides tracks for cells to migrate on. It binds signaling molecules, influencing their availability. And its physical properties, stiffness, elasticity, provide crucial cues to cells.
So the environment shapes the cells too.
Absolutely. You can see its importance in processes like gastrulation, where the basic body layers form, or neural tube formation. And now people are using synthetic ECMs with precisely controlled properties to dissect exactly how the matrix influences cell behavior and even cell fate decisions. Remodeling the ECM can actually tell a cell what to become.
And you mentioned forces earlier, the actual pushing and pulling.
Yes, tissue mechanics. This field has exploded recently. It's clear now that physical forces are not just consequences of development, they are actively instructive. They shape tissues and influence cell decisions.
How do forces get generated and sensed?
Force generation often comes from that actin-myosin network inside cells, the cell's muscles contracting. A key example is apical constriction, where cells squeeze their tops, causing an epithelial sheet to buckle inwards, forming tubes or pits, like in the developing gut or neural tube.
Cells squeezing themselves to make shapes.
Exactly. And how the tissue responds depends on its material properties. Is it stiff like a solid or fluid like honey? These properties change during development, too. Think about the zebrafish blastoderm spreading over the yolk that involves changes in tissue fluidity.
Okay. Any other surprising forces?
One that's getting more attention is hydrostatic pressure. Fluid pressure inside developing cavities, like the early blastocyst before implantation, or the developing inner ear. This pressure physically pushes on the surrounding cells and can drive changes in shape and size, like inflating a balloon.
Pressure as a shaping force. Cool. And cells can feel these forces.
Yes. That's mechanosensation. Cells have molecular centers that detect stretch, stiffness, shear stress. And these mechanical inputs can directly trigger signaling pathways inside the cell, changing gene expression and behavior.
Like feeling the environment and reacting.
Precisely. The classic example is how stem cell fate can be directed by matrix stiffness. Put stem cells on a soft matrix, they tend to become neurons. Intermediate stiffness, muscle, stiff matrix, bone.
Wow, the physical feel changes their destiny.
It's a powerful demonstration. So what's truly amazing, I think, is seeing how all these layers, the genetic instructions, the chemical signals flying between cells, and the physical forces pulling and pushing, all intricately interconnect and influence each other.
It's not just one thing, it's everything working together.
Exactly. It's this incredible dance of reciprocal causality, molecules influencing mechanics, mechanics feeding back onto gene expression, all unfolding to build an organism.
That incredible coordination from the tiny gene bursts right up to the large-scale forces shaping organs. It really drives home the complexity, but also the elegance of it all. And it makes you think. If we're starting to predict and even design these processes in computers, what does that imply about, individuality? Or what natural development even means?
Deep questions.
Which kind of brings us towards the end of this deep dive into developmental biology's revolution. We've covered a lot of ground from figuring out the molecular instructions to understanding the physical sculpting forces.
We really have. It's amazing to see how a field that started with careful observation has been utterly transformed by these waves of technology, genomics, imaging, computation. We can now genuinely aim for in toto biology, understand the whole system, from molecule to tissue to organism,
Yeah.
integrating across all those scales.
Yeah, that in toto idea is powerful. And the practical breakthroughs, like the sickle cell therapy we discussed, they feel like just the tip of the iceberg, don't they?
I think so. The review article we base this on speculates about the future. Things like virtual development, using AI to simulate and predict developmental outcomes with high fidelity. Or even, further down the line, designer organisms with capabilities not seen in nature.
Designer organisms.
Look,
That sounds like science fiction.
it's a long way off, maybe, but the trajectory points towards increasing control and understanding. Think about the potential to guide tissue regeneration, maybe even counteract some aspects of aging by manipulating developmental pathways or refining organoid technology to build better models or even replacement tissues.
The potential seems huge, but it also raises, well, big questions, right?
Absolutely. The ability to understand and potentially reshape human development using organoids or synthetic systems. It forces us to confront profound ethical questions. What does it mean to be human? Where should we draw the lines for intervention? These aren't just scientific questions, they're societal ones.
A conversation we definitely need to have.
For sure.
So maybe here's a final thought for everyone listening to mull over. If we really gain the ability to understand and manipulate all the fundamental processes that build a living being from one cell, every gene, every signal, every force, what does that truly mean for our future, for health, for development, maybe even for human evolution itself? What kinds of surprises and breakthroughs do you think are still waiting for us in this golden age? Something to ponder. Keep diving deep.